Hello, everyone. Welcome to a new episode of the Dissenter. I'm your host, Ricardo Lopes, and today I'm here with a very special guest, a return guest, Dr. Philip Ball. In our first interview, we talked about his book of minds, and I'm leaving a link to it in the description of this one. And today we're talking about his latest book, How Life Works, a user's guide to the new biology. So, Dr. Ball, welcome back to the show. It's always a huge pleasure to everyone.
Thanks for having me back, Ricardo. It's very nice.
So, since we're talking about life today, let me perhaps start with what I would think is a basic question, but perhaps one of the most complicated ones. So, what is life? I mean, is there any approach in, for example, physics or chemistry for us to be able to differentiate any sort of criteria to differentiate the organic from the inorganic, living things from non-living things, or not?
Well, if there is, no one has agreed on what that is. So I think it's fair to say that at the moment, there is no generally agreed definition of what life is. Which is amazing when you think about it. You know, it seems such a fundamental thing and such a kind of an obvious thing that surrounds us, but we cannot say scientifically exactly what it is that distinguishes a living thing from a non-living thing. So the approach I take in my book, I mean, fortunately, I don't have to answer that question, although I can't steer entirely away from it. But I'm talking about in the book about those things that we know are alive, and particularly about how our biology works. But in looking at that, one can't avoid that more general question: what is it that distinguishes, these living things? And one of the things that I suggest in the book is, in contrast to what often happens, is that you get these lists of what living things should require, these criteria. So they should be self-reproducing or self-replicating, they should have metabolism, maybe there's homeostasis and so on. All of those things are certainly properties that we find in living things, but the problem with lists like that is that we almost inevitably tend to find that there are systems that have at least some of those properties that we don't regard as living. Things like tornadoes, or things in the non-living world that have a kind of self-organizing capacity, but that we wouldn't regard as alive. And then perhaps there are some things that living things do that it seem to be excluded from a list like that. And it seems clear that there are these very few systems, in particular viruses, that seem to be right on the threshold of life. And there's no consensus about whether viruses are actually living systems or not. So what I end up doing in the book is to suggest that although this shouldn't be an absolute criterion of life, I think a more fruitful way to think about what it is that living systems have that makes them different from non-living is a notion called agency. And maybe we'll, get on to explore that idea in more detail later. But, in a nutshell, agency, it seems to me, it captures intuitively what we see living things do, which is that they have some capacity to change their environment and change themselves, most crucially, in order to achieve some particular goal. They have goals and purposes, and that's not something that we see in non-living systems, even complex ones like tornadoes. So it's that notion of goal-directedness that I think lies at the heart of life and of biology, and that we'll see sort of coming up again and again as we try to understand what makes life work.
So later we'll come back to agency, but I guess that perhaps there you might have been partially influenced by Dr. Kevin Mitchell through his book Free Agents or something like that, or not?
I absolutely drew on Kevin's work, his book Free Agents, which looks at what this notion might mean and how it evolved throughout time. It's a fabulous book. Kevin and I realized actually that we were both thinking about agency, at the same time, independently of one another. Kevin, I don't hesitate to say, Kevin has thought about this more deeply than I have and goes into this question more deeply than I have in his book. So it was a fabulous resource to me. But the two of us were thinking that this notion of agency seems to have some currency, and we weren't alone either. There are many other biologists and others, philosophers of biology, who are starting to think in terms of a notion of agency and are thinking, we really need to try to pin this down and ideally to get a theory of agency in order to understand life. So I think both of us are actually part of a broader movement that's going on, a controversial one, I should say, within biology to get to grips with this notion of agency.
But related to my first question, does it really matter, or do we really need to have a proper universal consensual definition of life? I mean, because wasn't it the case that we were able to learn things about how genes work and how genes relate to other molecules in our body and stuff like that, and things about different kinds of organisms without having a proper universal definition of what a gene or what a species is? And those are just two examples, I guess.
Yeah, well, I think it depends on the questions you want to ask. So if you're a biologist wanting to understand how the human body works or how bacteria work or how plants work or whatever, then you don't have to wrestle with that question because you know the subject of your investigations. You know that it's meaningful and has always been meaningful to think of it as being alive, as having this property. So you don't have to think about it more than that. I mean, I find that it's a little bit analogous to in quantum mechanics, how plenty of people, most physicists who use quantum mechanics don't have to think about what it actually means, which again is a question that actually we don't have any consensus about. They just get on and do the work. So, you can work this way in biology, but if you're someone like an astrobiologist who is searching for life on other worlds, then you have to have some sort of criterion for what you're looking for. And so, there it really does become important to really try to get to grips with, well, what is it that's characteristic of what we would recognize as a living thing, and how can we look for that in other worlds? And then you start to get into questions. I mean, going back to the definition that James Lovelock in the 1960s came up with for the Mars Viking, NASA's Viking missions, the lander missions, where they were wanting to, analyze the Martian soil and see if there were signs of life, and they needed to know what they were looking for. And Jim Lovelock suggested that a good criterion would be to look for quite extreme chemical disequilibrium, which means mixtures of chemicals that normally wouldn't be stable in that mixture. They would react and, reach some kind of equilibrium system. But if you see a strong disequilibrium, and that's what we see on the Earth, that's what we see in our atmosphere, in particular, for example, a mixture of lots of oxygen, which is a very reactive molecule, and methane and other molecules like that has just clearly been sustained, for a long period of time and hasn't settled down into an equilibrium. That is being driven by the biosphere. And so this was Jim's idea back in the 1960s, that's maybe what we should look for. We should look for chemical disequilibrium, but that's not enough. We recognize that's not enough because there are geological processes that can create that sort of disequilibrium over long periods. So that's a start for astrobiologists, but again, it's not enough. And that's the big challenge they now face now that we're able to go to other worlds and look at the atmospheres of other planets, even outside our solar system. What are we looking for if we're looking for life?
Mm-hmm. But of course, since life is such a complicated subject to talk about, many times we people, not just lay people, but even sometimes even the scientists themselves, resort to metaphors, like, for example, talking about living systems as machines, as computers, or something along those lines. And of course, throughout history, people have thought about life in different ways and through different metaphors. Do you think that these metaphors might be useful in some way, or are they just problematic? And if possible, we should just try to drop them because of the wrong ideas that they convey?
Well, I think that metaphors are essential. They're essential in
Okay.
science, and actually, I think they're essential in language generally. You know, we speak
Mhm.
in terms of metaphors, so they are a fundamental aspect of our thinking. So there's no getting away from them. And they are very useful. And, you know, even metaphors that, you know, were once used hundreds of years ago to understand maybe life or to understand complex systems that we now regard maybe as quite naive, they were still useful in their time.
Mhm.
I think the problem I perceive with some of these metaphors is that there is no mechanism for knowing when to dump them, when to get rid of them, recognizing when they are no longer useful. And, you know, it struck me in thinking about this, particularly in terms of life, that it's kind of peculiar that science is in this position. We have a mechanism, in theory at least anyway, a mechanism for getting rid of theories that are no longer useful because, you know, we test them against experiment. And if the theory and the experiment conflict often enough, we start to think maybe the theory needs changing. So that's fine. There's nothing like that with metaphors. And so metaphors can stick around and often do longer than they're welcome and longer than they're useful. And I think the idea that living organisms are kind of machines, to my mind, that seems to be one of these metaphors that has stuck around for too long. It's no longer helpful to think about it that way. Or rather, perhaps I should say, because I think it is nuanced, I think there are situations, certainly there are situations in biology where thinking of a component of a biological system, like a protein, for example, as a kind of machine, that makes absolute sense. And some of them look I mean, a few of them look remarkably like machines. So you have the bacteria, the bacterial flagellum, for example, which is a protein sort of assembly that allows these flagella, these sort of whip-like appendages, to spin round and let some bacteria swim. It looks just like a rotary motor in all sorts of ways, and so it makes sense to talk about it in that machine-like way. But when we think about how the whole organism works, the more closely we think about that, the less and less it looks like any sort of machine that we have ever made. It works in quite different ways. And so to my mind, the machine metaphor then starts to become not just inappropriate, but actually potentially misleading. And that, you know, that's been very controversial. There are some people who feel that actually thinking about us as machines, that's really a way of saying, I'm just a materialist. I don't think there's any sort of mystical, you know, vital spirit that's animating us. It's just atoms doing stuff. And, you know, that's I'm happy with that materialistic position myself. I'm happy to say that, you know, at the root of it all, if we look far enough down, all we see is atoms interacting. There's nothing mysterious beyond that. That's fine. But my problem is that when we start to look at the higher levels of how living systems work, they're not machine-like. So it's not a helpful metaphor anymore. So one of the things I wanted to try to do in my book is to think, well, what are the more useful metaphors that, you know, we now might want to call on in order to understand these things? And more broadly than that, what is the more useful kind of narrative we want to tell about how life works, rather than the conventional narrative, it seems to me, that certainly it's the one that we tend to hear about in the popular sphere, which is that it's all encoded in some sense in our DNA, in our genome. And life is simply, in essence, it's simply a readout of that information in the genome. So it's a computational metaphor. And again, there is some value for a computational metaphor of DNA, but if you think that is going to answer the question of what life is and how it works, then I'm afraid I think you're going to be mistaken.
So one of the alternatives in terms of the ways we could think about life that you present in your book is of living things as generators of meaning. What does that mean exactly?
Right. That comes again down to this notion that living things have goals and purposes. And it's kind of strange to me that this should be a controversial thing to say, because we have no. I think most people would have no argument with the fact that we have goals. That you had a goal in setting up this call today. Everything we do, we have some sort of goal that motivates us. We don't always perhaps quite know or articulate what those are, but we do. And we can see those, we can certainly see those in other animals. I think most people would think dogs have some sort of goal. Chimpanzees have some sort of goal. So it's not unique to us. And then it doesn't seem a big stretch to suggest that actually some kind of goal directedness goes, at least a long way, sort of through, I should say, rather than down, I think, but through the living world. In some sense, some plants have goals. And actually, I think it makes sense to see every living thing, down to single-celled bacteria, as in some sense having goals that they themselves determine in some sense. That doesn't mean to say that bacteria are thinking, what should I do now? Of course, but there is some goal directedness to it. So in order to achieve those goals, what any living system has to do, it's in some environment, it is constantly bombarded, if you like, with information or with things happening, things coming from that environment, how warm it is, what molecules are around, what other creatures are around, and so on. Living systems have sensory systems in order to take in that information, but they don't want to take it all in. It's too much. And most of that information, or a lot of that information, is simply not useful to it. So, for us, I mean, for example, the world, is also saturated with ultraviolet and infrared light that our visual system doesn't register. That of some creatures does, but ours doesn't. So that's information that, if you like, evolution has sort of found, is not sufficiently useful to us to develop a sensory system to make use of it. So, we ignore it and we don't even sort of recognize it in daily life, but it's absolutely out there. So what that really means is that there is some information out there that does not have meaning for us. It does not have value for us, value in the sense of being helpful to us in order to attain our goals. And this is what nature is constantly doing. For every organism, there are choices being made that essentially have been made by evolution. What matters in the environment? What do I have to attend to? And what can I ignore? And that's really what I mean by meaning here. So it's not meaning in any sort of deep metaphysical sense of, what's meaningful in my life. It is meaning in terms of each organism as an agent making value judgments about the information it receives. This matters and this doesn't. I'll attend to this and I won't attend to that. That's really something that, again, is quite distinctive to life, because it really is a kind of judgment of what matters and what doesn't, something that it's very hard to see how you could even have a sense of that in non-living systems. So that's what I mean by meaning, and I suggest in the book that actually that is one way of thinking about what life is. Life is a generator of meaning for the agents, the organisms that are living.
Mm-hmm. Yeah, I guess that some of the issues that some people might have, perhaps some of the old school philosophers of biology and evolutionary biologists, with the use of words like meaning and even perhaps in some contexts purpose and words like that, is that perhaps they would have a reaction where they would think, oh, so now we're getting into the religious realm or something like that. Are you saying that we are here for a reason, that we have purposes that are imbued into us as organisms, as living things, stuff like that? And even sometimes it also gets connected to ideas like teleology, that life follows a particular path, something like that. So I guess that maybe, at least for the more old school people, that's why they are a bit, I mean, they don't tend to like those kinds of words, I guess, because they associate them with that.
Yeah. There is a nervousness, I'd say, an allergy amongst some biologists to words like that, purpose and certainly to meaning, and also to goals. I was at a meeting in Minnesota a few weeks ago that was looking at all of these sorts of questions of agency and purpose, and there were biologists of all sorts of stamps and philosophers of biology there as well. So regular, biologists from university departments. And some of them were saying, one said in particular, the nice thing about a meeting like this is that we can voice these words that are taboo otherwise. They clearly felt, we need these concepts, but we're not allowed to say them. And it kind of seems crazy that we're not. And so there was a relief there that you could actually talk in these purposes. Now, some of that I think is perfectly understandable, because if you talk about things like purposes and goals, you could easily see how that could be weaponized really by creationists and people, interested in intelligent design, say, aha, so where does that purpose come from? It, must have had some divine source or whatever. But it really doesn't have to. You can naturalize these ideas, and I think that's part of the goal that's certainly part of the goal of getting a theory of agency, which has to include some notion of goal directedness. So, I don't think there has to be anything at all mystical in ideas like that. And I think it's also important to distinguish. You see, when goals and purpose are talked about, often for biologists, that seems to imply that evolution itself has some kind of direction, some kind of end point that it's heading towards. That's been an old idea that has sort of haunted evolutionary theory for a long time. And there is, as far as we can tell at the moment, there is absolutely no reason to believe that evolution has anything like a goal or a purpose. That it's famously described as this kind of blind search, I mean, not really a search for anything. It's a blind process that comes up with all these incredible sort of solutions. And there are some people who actually do ask whether there's some directionality to evolution. It's not an unreasonable question to ask. I'm pretty conservative on that question. I feel like, well, at the moment, I don't see any reason to suppose that is the case. But that's quite a different matter from saying the products of evolution, individual organisms themselves, not only can have goals and purposes, but that is in their intrinsic nature. That is what they are about. So there is no reason to link the two. You can have blind evolution producing systems, organisms that are goal driven. And in fact, I think that's exactly what evolution does.
Mm-hmm. We'll come back to organisms, but since we're also going to tackle here, at least to some extent, questions surrounding units and levels of selection in evolutionary biology, because that's one of the biggest questions out there. Let me start by asking you now about perhaps the most basic level that some people talk about, that is the level of the gene. So when it comes to genes, how do you think about their role and significance in life? And do you think that attaching words like instruction book, blueprint, or code to them is accurate enough or not?
Well, I think it's not accurate. I think it's actually very misleading. We still have this notion in sort of popular culture, because it's being pushed by biologists of various sorts, that the genome is our instruction book. Once you start to look at how we actually work and what genes do in that process, it seems to me that it makes no sense whatsoever, not just to me, actually, I think to a lot of biologists, it makes no sense whatsoever to think about it as an instruction book. It's not clear what that can possibly mean. You can't look, an instruction book, you can sort of read and you can understand, okay, this is how I need to put this thing together. There is no indication that we will ever be able to do anything like that from the genome, not least because it's clear that not all the information that is used, in a sense, to build the entire organism is simply in the genome. So I don't think that's a helpful way of looking at the genome, and in fact, I think it's an obstructive way and perhaps a dangerously misleading way to look at it, because it lies at the root of the cultural idea we have of genetic determinism, that what we are going to be is determined by our genes, which there are many ways that you can see that simply is not the case. I mean, to state an obvious one, the way our brains are wired up with its, I think, 86 billion neurons or something. There's lots of them, but the point is there are more of them, and certainly more connections between them than there are letters in the genome. There's not enough information there to specify how that's done. It can bias those connections in certain ways, and clearly does, because there are behavioral traits we have that we can correlate with what's in our genome, but it doesn't program how our brain is going to wire up. So, that's just one example. So the way I think it's more helpful, I mean, having said all that, I think it's absolutely essential to make clear that genes are clearly, they have a hugely central role in how we work. And not least because they are the thing, or at least the genome, or the information within the genome is the thing that is passed on between generations. So genes, as Richard Dawkins says, genes, I think, really do have a privileged status in evolutionary theory, because they are the thing that is passed between generations. But that doesn't mean to say they have to be the thing that contains all the information that is needed to put us together, because what are they passed on between? They're passed on between things that are already alive. They are, so that they are not. It's a little bit like, we tend to say, well, genes. I mean, in fact, Crick and Watson, when they discovered the structure of DNA in 1953, Jim Watson at least alleged that they were convinced that this was the secret of life. This was what life was all about. And yet, that gives rise to this peculiar notion that a living system plus its genes it is somehow, and it's it's still a living system. It has to be alive. The genome does nothing by itself. Put DNA in a beaker, even if you throw all the molecules that you think might be needed to put a system together, it does nothing. It's an inert molecule unless it's in a living system. And when it's in a living system, what it then does is provide resources, or the information for resources, in particular the information for building the proteins, the enzymes that catalyze our biochemical processes. But actually, we're also finding, and this is one of the key things I wanted to bring out in the book, we're also finding that there's an awful lot else in our genome besides instructions for making proteins. So, parts of our DNA, we've sort of really discovered only over the past 20 or 30 years how much this is true, that there are parts of DNA that don't encode proteins, but they do encode these RNA molecules that are like DNA, but they're just sort of shorter molecules. They're templated on DNA, and they used to be thought of pretty much all as being just the messenger that carries the information for making a protein from the DNA to the sort of machinery, and I'm going to use that term here, that makes the proteins. But some RNA molecules aren't like that. They're called non-coding RNA molecules, and they do things in their own right. They do kind of protein-like jobs, if you like. So there's more to the genome than protein coding genes. And in fact, there are also bits of the genome that don't encode proteins, but that are somehow involved in switching genes on and off. And they're dotted all over the genome. So the structure and the information that's in the genome is far more complex than just being a repository for protein coding genes plus a load of stuff that, doesn't really do anything useful at all. And the key point really is that the way all that information is used is contextual. What the cell does with it depends on all sorts of things, like what kind of cell it is. The way my liver cells use the information in my DNA is different from the way my skin cells do, even though it's the same DNA. So it's contextual. So the cell is integrating information from outside, even to the extent of deciding, if you like, which protein to make from a particular gene, because each protein coding gene doesn't just encode one protein, it can encode several. And in fact, on average, it encodes 6 different ones, because the information there can be sort of taken apart and stitched together in different ways to make different proteins. And the cell, if you like, decides which of those to make in a particular circumstance. So that's information coming from outside, even at the level of reading out a gene, which we thought was pretty sort of autonomous. So that's really what I mean, that it's really our genes and our DNA generally provide a very rich resource that the cell decides what it's going to use for.
Mm-hmm. And the fact that you mentioned at a certain point there that genes or DNA material just by itself is inert and it needs to be integrated into a living system to really be able to do anything. You mentioned there, for example, how genes that are part of cells in different kinds of tissues or organs, for example, their genes are expressed differently or produce different proteins depending on the type of tissue or organ they are part of. So, I mean, I guess that we have to look across different scales here to really understand what genes do, because it's the fact that they are part of a particular cell that has particular organelles and is receiving particular kinds of information from the cells around it and from different parts of the body, but also that it is part of a specific kind of tissue in a specific kind of organ, and also more generally that it is inside an organism that lives in a particular kind of environment, right? All of that matters.
Absolutely. So there is no privileged level in that system at which essentially life is happening, and it's certainly not at the level of the gene. I mean, this is the key thing to get across. I think genes are not alive. By the time if you want to understand how life works, you absolutely have to get down to the level of a gene to understand an aspect of that problem, but you're not going to answer it there because there is nothing alive at that level. And what really struck me as I was trying to find a way of articulating this is the difference between that picture, which is really a picture about developmental biology and how our day-to-day living works, the difference between that and the evolutionary picture. And in fact, there are plenty of people who feel that what we mean by gene is different if we're talking about a bit of DNA that does something in developmental terms, and if we're talking about this thing that in conventional evolutionary theory that people like Richard Dawkins have fantastically explained gets passed on between generations. They're not obviously the same thing. There is actually, just as with life, as you say, there is no consensus definition of what a gene actually is, but all of these different ideas are useful. And it really struck me that in the view, certainly the view famously brilliantly popularized by Richard Dawkins of the gene-centered view of evolution, he requires genes to be essentially like little agents themselves, all competing amongst each other in a kind of pool, undifferentiated pool of agents, where you lose sight of the fact that there are actually organisms here, and you literally do. So in the end, Richard Dawkins talks about a paradox of the fact that there are organisms at all, because why do you need them if all these genes are themselves little agents, sort of like little organisms competing and doing stuff? So that view requires us to put all the agency that is actually in living systems into the genes themselves. And, then the organisms themselves just become kind of passive vehicles that the genes build for their own purposes. So that's a view that can be useful for thinking about evolutionary biology, but is, I think, profoundly misleading in terms of thinking about what genes actually are as material things that contribute to, how cells and how organisms and tissues work. So that's really the distinction I'd want to make. And as you say, what a gene really does and means is contextual. It's using all this other information that's coming from elsewhere. And, that's the only way it can work. It's not entirely a sort of bottom-up process of simply reading out the genome in order to build the organism. The organism is constantly integrating all sorts of information. And one of the things that I found most fascinating in researching this book is how that integration of information happens even at the level of deciding whether to express, that is, whether to convert a given gene to its protein or not. It integrates all sorts of information, just to come up with, in the end, a binary decision. Should I turn this gene on or off, basically? Should I express it or not? And, it's a complex decision that involves many different molecules and actually often many different parts of our DNA, of the chromosome, in order to make that decision. So there is a very complex sort of set of principles that our biochemistry uses, our cells use, in order to make that decision. It's not something that is solely determined by what the genome itself contains.
And one very important thing here, I think, is that is the way we tend to relate genes to traits of a particular organism, right? Because we tend to have, or most people tend to have, this very simplistic idea of, so we have a gene and it produces a particular trait, or a group of genes that produce a particular trait. But with this new, more complex, but also more accurate view of how things work actually, in what ways do you think we need to reframe the way we think about the relationship between genes and traits?
Quite profoundly, but only in the sense, really, of making it more explicit in public what geneticists have pretty much always realized, because we've known for a long time that, for one thing, in general, a particular gene isn't involved in just a single trait. It often turns out to be involved in many different aspects of our physiology. And that's one of the things that makes, investigating genes so complicated. And in fact, that was one of the things that triggered me decades ago, actually, to start thinking, in these terms about whether we've got the right narrative. Because when I was working as an editor of Nature in the 1990s, the biological editors would come to our editorial meetings and they would say things like, I've got this paper that that we're going to publish that is, looking at this particular gene that we thought was involved in, I don't know, pregnancy or something. And it turns out that actually it's involved in something entirely different as well. And no one really seemed to have any idea of what this meant, why genes have all these different strings to their bow. And if we take a particular trait, we can't in general trace it back to a single gene. We find that there are many genes involved. And in some cases, if we look at something like how tall we are, there's clearly a strong genetic component to that. But it's spread over hundreds, maybe thousands of genes. Now, we only have 20,000 protein coding genes, or maybe even slightly fewer, in our bodies. So, if you've got a trait that involves 1,000 or a few thousand genes, that's kind of most of the protein coding genome, or, that's a significant proportion of it. And sometimes it really is most of it. So what that seems to be saying is that there are some traits that are kind of genome wide, they're influenced at a genome-wide level, and some researchers are talking now of a notion of omnigenics, of traits that seem to involve pretty much all our genes, or a huge proportion of them, some being more important than others, but most genes, pretty much all the genes, having only a tiny little influence themselves, in the sense that you see variations in the trait and variations in the sequence of that genome, but they account for hardly any of the actual variants by themselves. So that's really what we want what we need to get across. And as I say, that's something that's kind of always been recognized by geneticists, and yet somehow this kind of notion has grown up, that there is a gene for this, and a gene for this trait, a gene for that trait, a gene for, I don't know, boldness, a gene for, criminality or something. And we saw endless headlines like this in the 1990s, and you still see them now. And we seem to love this story in the media because it's a simple story to tell. We found the gene for X. And I slightly feel that geneticists now, they kind of tear their hair out every time one of these stories comes up, because they know that's not the real picture. And yet there seems to be a reluctance in the community to ask, well, why do we have this picture then? Who is it that has allowed this picture to develop? And I do think that geneticists and genetics generally is at least partly culpable for the fact that we have got that simplistic picture. So we need a different story to tell. And I think that once we recognize that genes are resources that cells use for doing various things, and resources that can do many different things, that are useful in many different ways, that is a better way to understand genes. So there are, for example, genes involved in our development that just keep turning up again and again in, a gene that is involved in the development of our fingers might turn up in also the development of our lungs, or something like this. There's a relatively small set of key developmental genes that seem to kind of somehow between themselves do seemingly almost everything in development. So, they're not genes for making a heart or making a lung or whatever. They're genes, general purpose developmental genes that our cells and our tissues are able to make use of in different contexts to do different jobs. So I think that it's a harder story to tell about what genes are, if we think of it this way. But that's why I think this notion of resources that are used by a higher level of the biological hierarchy, that is a more accurate picture of what they do.
Mm-hmm. And of course, we don't have time to get into all the details here, but people, of course, can read your book and learn more about it. But the ways by which, as we said earlier, the same gene can produce different proteins in different contexts. I mean, people also have to understand that, first of all, genes in different kinds of cells can be active or silent. And then they have to be transcribed. We have transcription, and they are transcribed into mRNA or messenger RNA. And then after that, there's post-transcription processes that can alter that mRNA before it goes into the ribosome to then get matched to a particular kind of tRNA and then produce the protein. So, it's not that we have a particular gene that gets transcribed and it goes directly connected to a particular kind of protein produced in the ribosome. Right.
No. Well, that's right. That's as I mentioned, a single gene in general can make several different proteins depending on the context. And the way that happens is that, and this was discovered in the 1970s, and it came out of nowhere, and people were astonished by it to begin with, that the sequence of sort of chemical letters in DNA is not the same as the sequence of chemical letters in proteins, that actually there are parts missing. And people realized that what happens is that so the DNA gets transcribed into the messenger RNA molecule, and then there's this big sort of assembly of molecules not just proteins, but actually also some of these non-coding RNAs that comes along called the spliceosome, which takes the mRNA, cuts it up, throws away some bits called introns, stitches together in various ways the other bits called exons, and that makes the template on which the protein is made. And so the spliceosome is really, if you like, making the choice about which protein you're going to make from a given mRNA, you know, the primary sort of transcript in terms of how it stitches it together. And again, that is contextual information. So it's integrating information, you know, of all sorts by bringing together all these different molecules, these proteins and non-coding RNAs, and somehow out of that comes this decision. So the more we've understood about that process, how the spliceosome works, you know, that's an aspect of this contextuality. And you know, that's just one sort of aspect, I guess, of, you know, how even at the level of the individual molecules that get made in our bodies, you know, how that involves higher level information. So really what this is, you could say challenging, is it's certainly challenging the conventional idea that information flow in biology is all bottom up from the gene to the proteins, and then somehow sort of magically the proteins, you know, or the information in the proteins sort of puts together us. It's challenging that. It's saying that information flow is coming in all different directions, and that idea, that old idea, is often sort of thought about in terms of what Francis Crick called the central dogma of DNA. And his idea was, you know, he was talking about how information gets from DNA to RNA to proteins. And in a sense, in a nutshell, he was saying once the information has got to the protein, it doesn't get back. There isn't a backward information flow from proteins to DNA. You don't get, you know, the protein doesn't sort of change the nature of the DNA itself. So in that sense, it's one directional, even though within that flow, there can be a little more complexity than that. That actually, you know, I think that if it's carefully stated, that's a pretty good summary of what actually happens at that level. There are some people who say, you know, that there are sort of particular processes that might challenge that idea. But on the whole, I think it's a fair summary of what happens between DNA and proteins. But as I say, the fact that the protein, you know, is not uniquely determined by the DNA means that there's actually more information coming in. It doesn't necessarily get back to DNA, but it's coming in at different levels of that process. And certainly what it doesn't mean is that this somehow extends to the whole organism. That actually, and this is really what I think Crick maybe to some extent had in mind, and definitely what James Watson had in mind when he kind of misstated repeatedly Crick's central dogma. Really, I think wanting to get the idea across that that information flow is all from the gene up to the organism, and it is this simple readout process. That's the message that has tended to sort of come across, and that's the one that I think is no longer tenable given what molecular biology has told us over the past several decades.
Mm-hmm. And talking about proteins, how important are proteins in life? I mean, because they seem to be everywhere and seem to participate in every single kind of different function out there. So how important are they?
Well, utterly crucial. I mean, they're extraordinary molecules. So I said that most proteins are enzymes of some sort. So basically, their proteins are strings of amino acids joined together, like beads on a necklace, really. And then, in general, the proteins will crumple up in water in the water of the cell into a particular shape, a compact shape, and that does its job. So the shape that it forms in a sense, that determines what the enzyme does. And the traditional idea was that within that shape, there's a little sort of binding pocket with a particular geometry that fits the molecule that the enzyme is meant to act on, like a lock and key. And that's really the root of the specificity that proteins have. So our cells are full of all these different proteins, thousands of different proteins, but because they have these very particular shapes, they only recognize their given target and kind of ignore everything else. That was the traditional idea, really. One of the things that I talk about in my book in relation to proteins, I mean, I should say that actually, you know, what I found was that in looking at all these different levels of how life works, pretty much in every case, over the past several decades, we've discovered something that significantly changes that picture. And one of the things that does so for proteins is that nice, neat idea of this sort of lock and key idea doesn't seem in general to be the case, that our protein, many of our proteins, perhaps a third, perhaps half, perhaps more, don't have these really, you know, tightly well-defined geometric shapes. They're actually floppy to some degree. They have what biochemists call disorder, and some of our proteins have a lot of it. They're really floppy, so they're not like tightly folded compact things. They've got all this kind of floppiness. And that is something, that protein disorder is something that seems to be crucial to the way a lot of those proteins work. One thing it does is if it has this floppiness, then it's no longer so particular about which molecules it binds to. It gives the proteins a kind of fairly generic stickiness and allow different proteins to stick together. And what it means is that different proteins can then find novel partners to stick to. And in that way, they can perhaps unite two completely different molecular pathways that, you know, responsible or that are implicated in different processes. And that is probably one of the things that lies at the heart of this polyfunctionality that genes have, because it's really, you know, it can be a polyfunctionality that the gene products, the proteins, have, because they stick to members of different sort of pathways and create crosstalk between them and create all this complexity in the network of interactions that allows information to be brought in from different sources. And we find that many of the most important sort of hub proteins in our own protein sort of networks, in our own biochemistry, are often ones that will have a fair degree of disorder. So they're good connectors between different aspects of our biochemistry, and this is what makes them so, you know, crucially important. So some of the proteins, for example, that seem to be implicated, or their genes are implicated in cancers, seem to be, you know, of this nature. They're somewhat disordered. So this notion of protein disorder is, you know, it's clearly very important for our biochemistry. And what it introduces, and this is just one way in which this concept is introduced, what it introduces is a kind of fuzziness to our molecular networks. Instead of being these precise chains of information transfer from this molecule to this molecule to this molecule, they're a bit fuzzy because the proteins themselves are a bit promiscuous in what they stick to. And it seems that this is actually an essential part of the way we work, because there is more of this fuzziness in us than there is in bacteria, which is ultimately what we were, if you like, descended from. So it seems that evolution has put it there on purpose. There is a value to it. And I think that the value, it seems to me, the value of it is that it allows this kind of contextuality, this kind of integration of many sources of information that is essential for an organism, a complex multicellular organism like us.
And of course, we're talking about genes and proteins, but in fact, what in the broader picture, what we have in an organism are networks, right? I mean, the sort of omics sciences like genomics, transcriptomics, proteomics, interactomics, because we have networks of genes, networks of transcription factors, networks of proteins, and then also networks are trying to understand how different kinds of molecules in a particular context interact with one another, right?
Yeah. And, you know, that too has been sort of recognized for a long time, that actually a key part of the answer of how we work is to try to understand these networks of interaction. But
Mhm.
what I wanted to where I wanted to shift that narrative is that in the past, those networks have tended to be thought of as a bit like the wiring circuitry in silicon chips. So they're a very precise path. This network has a very precise structure, and you can sort of
Mhm.
see, in biochemistry textbooks, you sort of see these diagrams of, these complex networks, but each molecule kind of speaks to this one or this one in very particular ways. What I wanted to get across is that those networks have this fuzziness. And so, I'm not even totally sure that network is, again, the right metaphor for
Okay.
thinking about them, because, or at least in general, because, another thing that happens is that, and this is something we've only discovered in the past sort of 20 years, is that cells make extensive use of loose collections of molecules rather than very particular sort of pairings, in these networks. They bring together clumps of molecules, of maybe hundreds of different molecules, in these sort of dense kind of liquid-like clusters, or some of them are liquid-like, they're sort of like gels actually, that form in transient ways in our cells. So we started to see, because we had the techniques to do it, all these little blobs and sort of speckles and things forming in our cells. It looks much more messy than we thought it was. And the more we understand about those, the more it seems that actually this is a mechanism that cells use that's really a physics-based mechanism. It's sort of analogous to the way vinegar separates from oil in salad dressing. It really is, like, in some cases at least, like a kind of liquid phase separation, a separation of one liquid phase from another. So, it's not some precise structure that where the molecules are joined together in certain ways, which is what we used to think. It's a loose, and I talk about it as a sort of committee of molecules that are often interacting with many others in these promiscuous ways. And yet out of them, and this is what committees are for, they're for making decisions. So out of them comes some kind of decision. So these structures, they're called biomolecular condensates, and they've been implicated in all kinds of cell processes. Often they're related to disease, but they're certainly implicated in the way genes are regulated, the way they're switched on and off. I mentioned that there are many molecules involved. They seem to be involved by forming one of these blobs, sort of basically on DNA within the cell nucleus, that through some kind of molecular, committee decides whether to turn the gene on or off. So if there are networks, often they're these fuzzy kind of blobby promiscuous networks rather than wiring diagrams.
Mhm. So I guess that now is a good time, since we've already talked about genes and proteins, to move one level up and talk about the cell. So how do you look at the cell in the broader context of an organism and in evolution?
Well, I think there's been, well, I know that there has been an increasing voice in biology, I think, over the past 20 years, to sort of reinstate the cell at the center of it. Some people have even talked about the rediscovery of the cell. Since the 1950s, it was all about the gene. And I think since, ironically, or perhaps actually not coincidentally, since the completion of the Human Genome Project, around the turn of the millennium, the cell has, been sort of returned to center stage. And I think, that is totally appropriate, because the cell is the minimal living thing. Below the level of the cell, obviously there are, crucial things happening that we need to understand, but nothing below that level, I think, is meaningfully alive. The cell is the unit of life. So in some sense, that is where it needs to start. And again, we found out things about what cells do and what they're capable of over the past sort of 2 decades or so that really overturn old ideas about that. And one of the most profound is we found out how, in what biologists, scientists generally call, how plastic the cell is, which really means how able it is to switch the state that it's in. So the old story was that our cells, we developed from a single-celled embryo, from a fertilized egg, And to begin with, those cells are said to be pluripotent. They're sort of like stem cells. They can make any tissue type in the body. And gradually, as the cells multiply, they get more and more specialized in different regions to become particular tissue types and ultimately to become particular organs to make the whole organism. And that's absolutely, true. That's the way things happen. We now see how contextual that is. We have better ways of following that developmental process, and we can see that cells sometimes, they seem to be following one path, and then they kind of switch and turn into a different tissue type because of the messages they've received from the cells around them. So again, just as molecules are like committees making decisions, so are cells. They're responding to all their neighbors in terms of deciding what kind of cell they're going to become, which is to say what the daughter cells, when cells divide, what sorts of cells they will be. And the thing that we have discovered, this was really in 2006, 2005, 2006, 2007, was that process, which used to be thought of as unidirectional from stem cell-like states to mature tissues where the cells are, skin cells or whatever, that actually is reversible. We can reverse the state of even of skin cells and turn them back into a stem cell-like state. That absolutely amazed biologists, even though there were some reasons to think there might be this sort of plasticity from older work. Really the ease with which that can be done. Basically, you have to just give skin cells you use a virus to bring into it some genes that are known to be active in stem cells. The reason I'm actually tapping my arm when I'm talking about this is that I've had this done to my own skin cells taken from my shoulder, which were turned by these techniques into a stem cell-like state and then grown into neurons. So skin into neurons. It was the most astonishing thing. And then those neurons start to organize themselves into things that look a little bit like brain-like structures, because cells seem to have that intrinsic knowledge of, once they become particular types of cell, they have, if you like, some intrinsic knowledge of how that kind of type of cell needs to be arranged and organized in the tissue in question. And so, the skin cells that would form sheets of skin, once they've been reprogrammed, and this is the terminology that's used, to be neurons, they start to create neuron-like structures, which look more like brain, like neural networks. So, this overturned our notion of what cells can do. And the whole sort of business of understanding how cells, it's called how cells acquire their fate, if you like, which sounds a bit doomy, but actually it just means what kind of tissue they make. This process of fate selection by cells, and how that involves genes and how that involves integrating information from around it as well in order to make that decision. That's another area that has really just boomed in recent times, and in ways that are fantastically useful, because if we can control cell fates and if we can grow cells into different tissue types, there are all sorts of possibilities for regenerative medicine and for tissue engineering. And we're starting to see some of those possibilities realized in medicine. So this is not just an aspect of, fundamentally understanding how we come into being. It's also something that has real medical applications.
So when earlier you said that you think of the cell as the unit of life, does that mean that you also think about it as the unit of selection in evolution, or not? And I mean, of course, I'm not trying to commit you to any particular kind of level of selection here. You can come from the paradigm of multi-level selection, and perhaps we can also come back to that when after we talk about tissues, organs, and bodies. But were you trying to imply that or not?
No, I wasn't trying to imply that. And in fact, to be honest, I was trying to duck that question. I have, the book ends, there's a sort of box section of text right at the end of the book, which says, which asks, what does all of this stuff mean for evolution? And I'd although I've had to talk about evolutionary processes at various times throughout the book, I was kind of putting that broad question to one side for two reasons. One is that I don't think anyone knows the answer to that yet, quite what the implications are for evolution of these more complex narratives and more complex ideas about the way we work. But certainly the second reason was that I know just enough about evolutionary theory to know how little I know. And to know how complex and subtle these evolutionary questions are. And so it's to understand what these ideas mean for evolutionary theory, it would require another entire book. And it's a book that certainly at this stage anyway could not be written by me, because I don't feel I have the knowledge to do so. So there is this big debate that's been going on for decades about what is the level of at which selection happens. And it's clearly complicated in the sense that ultimately selection for organisms happens at the level of the whole organism. The whole organism, either thrives or dies or is killed or whatever. So, it happens at that level. But what that means clearly is that if it dies, its genes, which is to say the particular variants of its that are in its genome, don't get passed on to progeny if it hasn't yet had any. So, that's the whole basis of natural selection, right? And clearly, as I say, because genes do have this privileged status, they're the thing that gets passed on to progeny. That's why the traditional idea is that selection happens at the level of the gene. And there have been all kinds of arguments from people who know far more about evolutionary theory than I do over the years of whether that's the whole story or whether there can be selection at other levels in biology. And particularly there was a big argument between Richard Dawkins and E.O. Wilson about the idea of group selection, of whether selection happens not just at the level of individual organisms, but there are things that are good for the group that are sort of transferred in the genome. I think the fact that those arguments are happening is a reflection of how subtle these questions are, and they're not resolved yet. So that's really all I felt I could say about the implications for evolutionary biology, to recognize that there are these still open questions, and that I think it's for others who know more about the subject than me to try to integrate them into this new picture of how life works.
Fair enough. So let me ask you now a little bit about tissues, organs, and bodies. So how does a cell know that it should grow, that it should replicate, and when to stop? Because this is also very relevant, for example, to things like understanding how tumors and cancers grow, because that's basically it starts from one single cell not being able to stop replicating, right?
Yeah. Well, I mean, the answer to that question is one of the reasons why I think it doesn't really make any sense in developmental terms to think of our development as a simple readout from the genome, because those decisions are clearly crucial. How does an organ know when to stop? Our organs grow to a certain size and then they stop. How do they know that? It's not programmed into there's no gene that is kind of suddenly kicks in and stops that process at a certain stage. It's contextual information made at the level of the tissue. So one way in which, for example, it's really about how cells communicate with one another to know what they're up to. That's really, I think that's the right way to kind of think about it. They have to know, how many neighbors have I got? How big has this whole sort of tissue got? And they communicate in general in three ways. One is chemical. They send out things like hormones that, get sort of plug into proteins at the surface of other cells and create signals that go down to the genome and so forth. One is electrical. So all cells, not just our neurons, but actually all cells in general have the potential for making electrical connections with others that kind of sense the gradients in voltage really and potential across cell membranes, and that seems to be a control mechanism. And one of the most crucial is mechanical. So cells, if you just sort of tug on a cell, you can change its cell fate. You can change the kind of cell that it will turn into if you tug on a stem cell. And that's why if you grow stem cells on a soft surface, they might develop in a different direction than if you grow them on a hard surface, because they're responding to the tensions created by adhesion to those different surfaces. So they respond to mechanical signals. And so, for example, that's how it is that if a fetus is growing with some kind of, let's say, some kind of developmental defect that means maybe a limb is shorter than it might normally be, it's kind of, when you think about it, you think, well, how do all the different tissues of that limb know to stop growing? If your bone isn't growing enough, okay, that's one thing, but then why do all the other tissues respond? Well, one way it happens is that as the skin is growing, the cells are sensing the tension in that skin, and if the skin has become too taut, then the cells know they need to continue proliferating and release that tension. If it's become too loose, then they will stop growing. So there's that mechanical sensing that happens at the tissue level that is determining things like the overall morphology of the organism. And that, I don't see that as, in any meaningful sense encoded in the genome, because it's entirely contextual. It depends on what the other things around it are doing. And sometimes it might depend on environmental signals outside the whole organism. So, this is another sort of reflection of how at each level of the biological hierarchy, there are rules that are governing the growth and the morphology that are drawing on genetic resources, and they're perhaps feeding back to genetic resources, so that the cells stopping growing means that there's something happening at the level of the genome, but that are actually using contextual information from the environment rather than simply following a program.
Okay, but it's one thing for cells and tissues to know when to grow, for cells to know when to replicate or when to stop replicating. But how is it that, or why is it that organs have particular kinds of shapes and also bodies have particular kinds of shapes? I mean, how do we go from cells to tissues and then to particular kinds of shapes of organs and entire organisms?
Yeah, well, I mean, that really, that question is the whole of developmental biology, which is, a huge field and still unfolding, and we don't understand the answers to all those questions. But we do have answers in some specific cases. So, for example, if we think about how the lung grows, it's a branching process, and one that has lots of similarity, not just in terms of how the thing looks, but perhaps in terms of the basic physics of that process with the growth of a tree or the growth of a river network. The branches grow and then they maybe split, and they grow off and they split again. And that's a sort of complex process, a sort of feedback process, really. So, for example, the same is true of our system of blood vessels, and that's really about at the finest level of capillaries. That's contingent. It's like the way I was talking about our brain wires up. There's nothing programmed in that. It's cells making decisions on the hoof about what's needed. And the way that happens is that if a tissue doesn't have an adequate blood supply, so there are no nearby capillaries and it's not getting oxygen, the cells kind of send out a distress signal, a chemical distress signal. They change their state into this state that is, creating this signal, and it's basically a signal that tells the blood vessel forming cells, come over here. And they will, they'll start to proliferate, towards that region of oxygen depletion and create a new blood vessel that way. So it's entirely sort of contingent, and it's kind of improvising, I suppose, as it goes. But then there are other developmental processes that are more that are more, you could say, more deterministic in that they will turn out a certain way unless something goes wrong. In general, we all have 5 fingers, for example, but there is no gene that prescribes that. There is no gene for the fiveness of our fingers or toes. The way that process works is, and this is a beautiful example of the kind of autonomous processes that happen at that developmental level, that we now think that it's related to a process that was first talked about theoretically by the mathematician Alan Turing in the 1950s, where Turing showed that how it's possible, and he was interested in, he asked this question because he was interested in development too. He showed that it was possible for particular mixtures of different chemical agents, if they have particular properties of how they react with themselves and with each other. In some circumstances, you can start off with a totally uniform mixture of them, and they will spontaneously separate into regions with different concentrations of the different amounts, so into a kind of patchiness. And the generic forms that tend to arise from that so-called Turing process are spots and stripes. So like sort of zebra stripe type patterns, or more or less randomly sort of positioned spots. And we see both of those processes in biology, and it seems that this Turing process is really used by biology. So the spotlight processes you can see in our hair follicles, for example, that the little pores and the follicles that make the hairs, they're like these sort of spotty patterns, and we think that seems to be a Turing-like process. Our fingers are stripes. They're Turing stripes. So as the limb bud develops, it starts off as a sort of paddle, an undifferentiated paddle. But there is an interaction between some of these developmental proteins that I talked about earlier on that causes the appearance of stripes, within which bone forming genes are switched on. So you start to get the bones forming, and then in the tissue in between, the cells die away. And that's why there's nothing there, and that's why sometimes, if that doesn't happen fully, you get sort of webbed skin, that sometimes happens between fingers or toes. So that's how that process is working. It's drawing again on genetically encoded developmental proteins, but they're proteins that do all sorts of other things as well. And the actual process that is creating this structure is one it's a higher level process that's kind of self-organizing itself into these structures.
So earlier in our conversation, we talked about words and things like meaning, purpose, goals, but I think that we left out of that discussion, or we saved for later, a discussion surrounding the term agent and what agency means. So what is an agent in biological terms? And I mean, at what level can we see agency? I mean, can, for example, a single-celled organism already be an agent, or does it need to be more complex than that?
Well, again, it's a word that doesn't have any generally agreed meaning, and it certainly differs in meaning across different scientific disciplines. So there are some, I guess, maybe in psychology, who would only recognize agency as something that involves intention and that involves, sort of conscious cognition. That's, an expression of our human agency. Within engineering, there is can be a meaning of agency that doesn't necessarily correlate with the one in biology. So it depends who you ask. But the way I talk about it in the book, and the way that I think is perhaps it feels the most meaningful way to think about it in biology, is agency is the ability of some entity, of an organism, basically, to it's the ability of an agent, let's leave it general like that, to set itself goals, to have self-determined goals, and to manipulate its environment and perhaps itself in order to attain those goals. An entity that can do that is an agent.
But then that sort of entity could be a single-celled organism.
I think so. Yes, I would absolutely. Well, in fact, I would absolutely say it can be that. There's plenty of good reasons now, good experimental evidence to see that single cells, bacteria, or even single cells of our own tissues have a kind of agency. For example, you can in cell cultures of bacterial cells or our own cells, they are nominally, you can grow them so that they're nominally identical cells. They're genetically identical. They've had the same kind of history as far as you can tell, and yet you can provide those cells with some kind of stimulus, maybe heat or maybe some chemical, and they won't all respond in the same way, because there are maybe just sort of random or maybe non-random, because of where they are in the colony, differences that have arisen between them that have created differences in the internal state of those cells, which genes they're expressing, which molecules they're making, and those are and that internal state is an aspect of the decision that each cell makes in order to about responding to this stimulus. So that's a characteristic of agents, that they're not just sort of autonomous, that automatically you give them a particular stimulus and they'll respond in a certain way. There's a degree to which that is true, certainly for single cells, but we know that it's not always true, that it depends also on the internal state of the agent. And this is one of the, you mentioned Kevin Mitchell's book on agency, and Kevin has a nice, in fact, it's work that he did with his student Henry Potter, where they have a nice breakdown of characteristics that agents have, and this dependence on internal states is one of them. And what that tells you is that an agent has to have some internal complexity in order to really qualify as an agent, and that's why a gene can't, because it simply doesn't, and a protein really doesn't. There are some proteins that are sort of big enough and complex enough that it's meaningful to ask that question, are they starting to show some sort of agency? But in general, you only will find that in something that has the internal complexity of something like a cell. So that's where I feel agency, as well as actual life, really begins.
So in your book, at a certain point, you also talk about and comment on medical approaches to biology. So could you tell us a little bit about that? And when it comes to the sort of paradigm that is the most dominant nowadays in medicine, what do you think about it?
Well, I think one of the messages for medicine that might come out of the kinds of things I consider in the book is, because of these this hierarchy of levels in the way life works, each of which has some degree of autonomy, some degree of independence from the details of what's happening at the lower levels. What that really means is there's a that causation, what is really causing, if you like, if you're thinking about the shape, the structure, the behavior of the whole system, what is really causing that, it might arise at many, perhaps any of those levels. It's not a cause that's all determined at the genetic level. Some things are. And if we're thinking now about disease, if we think about something like cystic fibrosis, there is a particular gene where there are several mutant varieties of that gene that some people inherit. And, if you have them, then you are likely to get cystic fibrosis. Not actually completely, it doesn't necessarily do you to having that. It depends. There's still some dependence on higher levels that can compensate. But, if you have cystic fibrosis, you pretty much certainly have one of these gene variants. So in that case, it's meaningful to think of that gene as being a real cause of the disease. But many other diseases, because I talked about the way that our traits are spread throughout the genome, so the common the most common diseases that we face, things like, heart disease, and or cardiovascular problems generally, obesity, diabetes, they're spread throughout the genome, and so causation can't in any meaningful sense certainly be ascribed to a single gene, but I think it can't really be ascribed to the genome itself. It's things that are happening at the physiological level that are being affected by genetics, but the causation is perhaps happening at a higher level. If we think about COVID, for example, the people who had serious reactions to an infection by the COVID virus, and many of the people who died from it, that the problem there was how their immune system was responding. That it was a sort of often an over response of the immune system that caused all kinds of problems with tissues, all kinds of respiratory problems. It seems that there is a genetic aspect to that propensity, and we're only starting to uncover that now. But really, the causative level of the problem there was at the level of the immune system, and so that was where often we tried to intervene. That some of the drugs that were most useful were anti-inflammatories that calmed down the immune system. And this is the general message then of the book, that really, if we have this multi-level causation in biology, if we're wanting to put something right that has gone wrong at the level of the organism, at the level of the body, then it makes sense to try to identify, where is the primary causation arising for this particular condition. So the predominant idea, over since certainly it was the Human Genome Project was partly predicated on this. Once we understand the genome, we'll be able to figure out which genes are responsible for pretty much any disease, and we can go in and do something about it. But if causation is not rooted in the genes, that's not going to be very useful. And that's we've seen that the Genome Project has been fantastically useful as a resource for all sorts of reasons, particularly for identifying people who might have a genetic propensity or vulnerability to disease, but it has produced really very few actual cures, because that's perhaps not the level at which cure will be found. So that's really the general message, that and I think that's particularly been notable in cancer. And there are many people now saying we've had years of of this notion that we'll identify the cancer genes and that will allow us to stop cancer, and it hasn't worked. And maybe we need to rethink it. And perhaps part of that rethinking is to think about cancer more at the systemic level, more at perhaps the level of a dysfunction of cells or even of tissues, rather than of genes.
So if I understand it correctly, then the main proposal here for doctors, for researchers in medicine, would be for them to reframe the way they think about causation in disease. Or not?
I think so, and I wouldn't want to claim that this is some bold idea that I've come up with, that I'm sort of telling the medical community. I don't mean to say that. I think that this is something that actually people are expressing more and more. Anyway, I mean, as I say, in particular in the case of cancer, where the genetic approaches have been really, frankly, so disappointing. I mean, I talked earlier about having had cancer myself, prostate cancer, recently, and it's a stark reminder of how we actually generally deal with cancer still, which is high-tech medieval, really. That it's in my case, a robot goes in and performs surgery and cuts out the bit that has the cancer in, and it's fantastic that it worked, it can work, and that it worked in my case, but that's still the level that we're at. We just cut it out, or we pump ourselves full of toxic stuff and hope that it kills the cancer cells faster than it kills all our healthy cells. So we still generally relying on these crude methods rather than anything finely sort of targeted at particular genes. So I think we're already discovering, we're already realizing that actually that we need to take this broader view of medicine than just sort of thinking that you can address everything at the genetic level. And, again, thinking about cancer, the immune system seems to be a conduit for all kinds of, there's hardly a disease that we know of that doesn't involve the immune system in some way. And it's really striking that the immune system is now one of the areas that we're targeting for dealing with cancer. Immunotherapy is very effective for particular types of cancer. It doesn't work by any means for all cancers, but for some, it seems to show a great deal of promise to sort of supercharge our immune system to deal with the cancer. So in a way, that's kind of helping the body at the right level to deal with the problem itself, rather than us kind of going in there and trying to do microsurgery on genes.
Mhm. But going back to when we talked about how context is so important to understand how life works, in the case of, for example, studying and treating cancers, don't you think that perhaps we should need more personalized medicine here? Because it's not just that every cancer is different from the others, but also I would imagine that in different people, even the same type of cancer would be different, right?
Yeah, absolutely. And I think that that's the kind of personalized medicine that I think really does hold a lot of promise. Because as you say, it's now recognized, because we have the techniques to look in detail at cancers, it's now recognized that in a sense, every cancer is individual, and it's not just cells proliferating sort of wildly and indiscriminately. Actually, a lot of tumors look more like, I quoted in the book, I think, that it's like a deranged development, that actually they have different types of cell within the tumor, different types of cancer cell that look like, they're differentiating in much the same way that they do in normal development. And if we understand what different types of cell are in a particular tumor, we can then quite possibly target that tumor with the right kind of treatment, the right kind of drugs for that particular tumor. And even, as you say, in a single person, one tumor might not be the same as another. So absolutely. And I think, that's one area where being able to genetically analyze the cells that are in a tumor is absolutely valuable. That tells you, that's really valuable information for the types of cell it is and the types of treatment that it might respond to. And I think it's very striking that, a lot of people who are talking about personalized medicine now, we got this idea, I think, often sort of pushed to us. So what that means is we'll have our genomes profiled, and then our genomes will tell us what treatments are best for us. That there might be some degree that will happen, but I think people generally now in personalized medicine recognize this hierarchy of levels, and that actually what we also need to do is to be profiling ourselves at the tissue level, at the cell level, at the physiological level, to understand, what treatment is going to be best for the individual. So personalized medicine is coming around to this a kind of holistic picture, I think, of understanding the biology, understanding how life works at all these different levels, so that we can best understand how to intervene for an individual.
So one last topic then. Toward the end of your book, you go beyond talking about how life works, and you also talk about redesigning life. So as we accumulate knowledge about how life works, what do you think would be some of the main ways we could redesign life?
Well, I've touched on one of them already, which is this notion of cell reprogramming for tissue regeneration. And one way in which that's developing is that I said when I had my skin cells reprogrammed to neurons, they started making something that looked a little bit like a sort of brain-like structure. That's a structure called an organoid. So it's grown in a dish, and it starts to, in some crude way, to resemble a sort of embryonic brain. If you reprogram the cells not to become neurons, but to become some other tissue type, then they start to assemble into structures that look a bit like the respective organ. If you program them to become like the epithelium of the gut, they start to make, a tube-like, gut-like structure with all these little sort of protrusions that we have in our gut. If you make them into pancreatic cells, they will start to become like a little pancreas. So these so-called organoids, they can be used as models to understand to fundamentally investigate the structure and the properties and how these organs will respond to particular treatments. So they can help us understand the developmental process in a dish. But they can also be potentially grown into structures that could be used that could be reimplanted in the body. So people are interested in, for example, repairing neural damage by growing neural structures in a dish and then perhaps implanting that neural tissue into the body, or doing that with muscle or whatever. And there are approaches that are starting to pay off in this way. Or we can, in some cases, we can think about reprogramming cells directly within the body rather than having to take them out and grow them in a dish. There are complications to that process, but in some ways there are also advantages. You want, if you're regrowing neural tissue, you want it to be integrated into the neurons that are already there, which can happen much more easily if it's growing in situ rather than grown in a dish and sort of grafted back in. So, those sorts of possibilities are really extraordinary for regenerative medicine. But beyond that, I think once we start to recognize how the developmental process really goes on and how cells acquire fates and communicate with each other, it opens up questions that almost seem verging on the philosophical of, what does that mean about the range of different morphologies that the human body could have, or that tissues generally could have? You can ask questions like, well, could there be states that cells could form because they're, these incredibly complex systems with, thousands of different proteins, but they only form a limited number of tissues in our body. It seems unlikely that those are the only things cells could form. Could there be tissue types that that don't appear in our body, but that could actually be useful, could actually, do something? Could they grow into, could we program cells to grow into different shapes? Can we understand these developmental processes well enough that we can make not just a little organoid, but actually a group of different organoids that, start communicating with one another like a system of organs? I mean, once you start thinking about down this line, the possibilities become mind-boggling and also potentially grotesque. Could you grow a kind of new sort of organ or new organism or mini organism where, several of these organoids have kind of wired up? We've already, researchers have already been able to wire up a brain organoid to a bit of muscle tissue so that the signals from the brain can make the muscle do things. How far along those lines can we go? What are the ethical considerations about that in particular? And, particularly when you're thinking about growing brain organoids, the more brain-like they become, the better model they are for understanding how the real brain works, and the bigger they become, the better model they are. But also, the more you have to then start thinking, is there some point, if we're growing them from human cells, some point at which we have to start thinking, could this thing be conscious? What does that mean? What does it mean for a bit of, isolated brain-like tissue outside the body to have consciousness? Could it, in any sense, have feeling? We don't know the answer to these questions, but we absolutely have to ask them once we start growing these things. So, yeah, in terms of being able to understand and control and direct developmental processes, including ones outside the body, incredible possibilities open up for growing new kinds of, living mechanism, or, possibly new kinds of, organ. But with them come some really quite tricky ethical and also philosophical questions.
And I would imagine that particularly if in the future we accumulate enough knowledge to be able to create completely new life forms, that could also raise some ethical questions, like, for example, if we were to create a new life form that is sentient, then how would we treat it? How would we deal with it? In what ways would we use it if that was the case? And also, if it was something that we would then be releasing into a particular ecosystem, then we should also care about how it would interact with other kinds of organisms there.
Yeah, yeah.
It could disrupt the ecosystem. Right?
Absolutely, yes. Well, these are already the sorts of questions that people working in synthetic biology, have to understand. And in general, this idea of synthetic biology, of redesigning living organisms for particular purposes, in general, that's been confined to doing things to bacteria, because bacteria are simpler than us. They're more predictable than us. You can insert particular genes into them, and we've actually been doing this in the 1970s to get them to make particular chemicals, or to alter the ways they behave in quite well defined ways. And there can be fantastic uses for this, for making new sort of biomaterials and so on, or perhaps even programming bacteria to be environmental remediators, to eat up lots of pollution or something. But if you're doing even at that level, you have to start thinking, well, what are the implications of releasing something engineered like this into the natural environment where it can potentially evolve? But once you start to think about multicellular synthetic biology, which is still relatively new because it's much harder to do, because you then have to think about more levels of the hierarchy and how we can control them, then these broader questions arise. And if you're thinking about doing that with human cells, or for example with perhaps mixtures of human cells and those of other animals, or those of other organisms, what is the nature of the entity that you're creating? Is it in some sense a new kind of living entity? What does that mean? What are the safety considerations and so on? So these kinds of considerations are going to become more and more complex. I mean, one area that I just also touch on in the book is that in understanding human embryology, we can now create out of stem cells structures that look in many respects like human embryos, that have not been created by any eggs or sperm being involved at all. They're just taking stem cells, and the stem cells will organize themselves if you do it in the right way and give them the right sort of tissues and nutrients. They will organize themselves into something that looks like a human embryo, and that can be really useful as a kind of whole embryo organoid, really, for understanding development. But of course the ethical questions there become even more profound. And should we treat it as we do actual human embryos, with the same kind of rules governing what can be done? Or is it a different sort? Is it a tissue culture? Is it a different sort of organism? Really, we don't know the answer to these things, but they're ones that people are now having to really sort of grapple with.
Mm-hmm. Okay, so let's wrap up the interview here. The book is again How Life Works, a user's guide to the new biology. I'm leaving a link to it in the description down below. And Dr. Ball, would you like to tell people again where they can find you and your work on the internet?
Well, yes, I guess you can. I mean, I have a website which I guess you can put up, Ricardo, www.flipboard.co.uk. Can Google me and you'll probably find me, and a full list of my books is on that website.
By the way, could we expect a new book somewhere in the future, or?
Yes, inevitably, I guess. I'm not going to say too much about the I have to admit, rather guiltily, I'm working on, in one way or another, on 3 books at the moment. And I'm not going to say too much about them, except that they're underway, except for one, which is going to be published, I think, hopefully later this year. And it's an illustrated history of alchemy. It's working with a publisher who produces beautiful illustrated books that I've done a couple before with them. They always look fantastic. And yes, that's basically what it's about. It's trying to reposition the way we think about alchemy as something that's not just some kind of weird superstition, but is actually an important part of the history of science.
Great. So I'm very much looking forward to it. And thank you so much again for taking the time to come on the show. It's always a pleasure to talk with you.
Well, it's a pleasure for me too. Thanks very much, Ricardo.
Hi guys, thank you for watching this interview until the end. If you liked it, please share it, leave a like and hit the subscription button. This show is brought to you by Enlights, learning and development done differently. Check their website at enlights.com, and also please consider supporting the show on Patreon or PayPal. I would also like to give a huge thank you to my main patrons and PayPal supporters: Per Ergo Larsen, Jerry Mullerens, Frederick Sunda, Bernardo Seixas, Olaf Alex, Adam Kessel, Matthew Whittingbourd, Arno Wolf, Tim Hollis, Eric Alenia, John Connors, Philip Forrest Connolly, Dan Demetri, Robert Windiger, Rui Inácio, Zup, Marco Neves, Colin Holbrook, Phil Kavanagh, Michael Stormir, Samuel André, Francis Forte, Tiago Nunes, Fergal Cossen, Hal Herzog, Nuno Machado, Jonathan Labyrinth, João Linhares, Stantantia, Samuel Correia, Eric Hines, Mark Smith, João Eira, Tom Hummel, Sardas France, David Sloan Wilson, Yacila Dez Araújo, Romain Roach, Diego Londonho Correia, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavlo Stasevski, Nelek Bach, Guy Madison, Gary G. Alman, Sam Afzal, Adrian Yeager, Paul Tolentino, João Barbosa, Julian Price, Edward Hall, Edin Bronner, Douglas Fry, Franca Bortolotti, Gabriel Ponce Cortez, Ursula Litsky, Scott, Zachary Fish, Tim Duffy, Sonny Smith, John Wiseman, Daniel Friedman, William Buckner, Paul George Arnaud, Luke Loaki, Georgios Theophanos, Chris Williamson, Peter Wolozin, David Williams, Diogo Costa, Anton Erickson, Charles Murray, Alex Shaw, Maury Martinez, Coralie Chevalier, Bangalore Atheists, Larry Dealey Jr., Old Erringbone, Sterry, Michael Bailey, Dan Sperber, Robert Grassi, Igor N., Jeff McMahon, Jake Zool, Barnabas Radix, Mark Campbell, Thomas Dovner, Luke Nissen, Chris Story, Kimberly Johnson, Benjamin Galbert, Jessica Nowicki, Linda Brandon, Nicholas Carlson, Ismail Benzliman, George Coriatis, Valentin Steinman, Per Crowley, Kate Van Goller, Alexander Hubbard, Liam Dunaway, BR, Masood Ali Mohammadi, Perpendicular, Jonas Hertner, Ursula Goodenough, Gregory Hastings, David Pinsoff, Sean Nelson, Mike Lavigne, and Dios Necht. A special thanks to my producers: Isar Webb, Jim Frank, Lucas Stafiniak, Tom Vanagdam, Bernard Igni, Curtis Dixon, Benedict Mueller, Thomas Trumbull, Catherine and Patrick Tobin, Giancarlo Montenegro, Alnick Ortiz, and Nick Golden, and to my executive producers Matthew Lavender, Sergio Quadriano, Bogdan Kanivets, and Rosie. Thank you for all.